and they're going to share with us the work that's happening at the Rare Genomes Project. Grace is a clinical project manager for the Rare Genomes Project. She's a licensed and board certified genetic counselor with a background in pediatric rare disease, gene discovery, and genomic sequencing. And Brian is a clinical project coordinator and focuses on family engagement with the study and outreach to the rare disease community. He has a background in neurobiology and patient advocacy. So we thank you guys for being with us today. We really look forward to hearing what you have to say. And I do want to share with everyone that I think we have about a quarter of the people with us today are families or family advocates, and then the rest of us are providers or other types of professionals. But we always really value and enjoy the fact that we have families joining us to learn alongside and together. So with that, I will hand it off to Grace and Brian.
Are you going to wear that?
Yep. Yes, except I'm going to put shoes on.
That's the shoes you're going to wear?
No, I'm going to put shoes on.
Dr. David, can you mute yourself?
Am I going to wear this tonight?
Shh, shh, shh.
What do you?
Thank you. Sorry.
That's okay. Hi, everyone. Thank you for such a nice introduction, Alyssa. Let me see if there's a way. We have some new technology in the room that we're presenting, and there we go. Great. So I can see the slides and your faces. So I think we are all set to go. So I'm Grace.
I'm Brian.
And as you mentioned, we work with a research project here at the Broad Institute called the Rare Genomes Project. It's nice to see some familiar names on this call. I recognize some genetic counselors and other providers who have referred families to us already. And we're excited to kind of connect with those of you who we haven't interacted with before. So thank you for having us. We also have a couple other team members joining us to listen in today. So shout out to Melanie, another genetic counselor on the project, and then Eva, Jillian, and Lale, who are all research coordinators for the project as well, who are joining too. So I'll take it away and get started. Our plan is to give you some background about our project and give you an explanation of kind of which kinds of families we are working with. If you are a provider who sees patients, we hope we give you an idea of which families would be a strong fit for our project. If you're a family yourself, this is a project that you can self-apply for, so you do not need a provider referral. So if you feel like your family meets eligibility criteria, you're welcome to fill out the application online, and we'll talk more about that. But just to kind of set the stage, and we're definitely going to make sure that we leave time for questions at the end. So if questions do pop up, maybe just jot them down so that they stick with you. And if we haven't answered your question by the end, then please, we'll invite folks to unmute and jump in and ask those questions once we've gotten through the slides. That sounds good. All right. So I'll start with just telling you a little bit about our team, because we have a pretty great team behind us. So our team is embedded within a larger rare disease research group at the Broad Institute. You can see actually a pretty recent picture of a number of us there. So we are a pretty diverse team in terms of our expertise and backgrounds. We have both clinical and molecular geneticists, genetic counselors, variant analysts. We have a software engineering team, bioinformaticians, statistical geneticists. And because we have such a diverse range of expertise, we're able to be really collaborative and come up with research tools that really help us dig into finding diagnoses for patients who are on a diagnostic odyssey and have been undiagnosed until now. So we also, outside of the Rare Genomes Project, create community resources for the genomics community, things like Nomad, ClinVar, and ClinGen, for those of you who are familiar with those resources. All right. So this slide we like to kind of start with because it kind of drives everything that we do here at the Rare Genomes Project. We know that every diagnosis can make a difference for a family, and different families will have experience different impacts if and when they receive a diagnosis, but we're aware that there are many different motivations for seeking a diagnosis. Some families are really invested in prognosis information, and those are questions that we might hear about, kind of what will the future hold for my child or myself. Lots of folks are really interested in whether or not there could be a change in medical care, like increased screening or actually the removal of unnecessary screening or visits. All are quite hopeful for treatment and cures. I think, we know that in the genomic medicine space, there's still only a very small fraction of conditions that are amenable to a specific therapy or medication, but that number is growing and relatively rapidly. So that particular impact I anticipate will be actually more significant as time moves forward. There's also the psychosocial aspect that we're aware of, the psychological relief of understanding why a particular genetic condition has occurred, of kind of knowing what the road might hold, and also finding community. So we love resources like NIMAC, and we love patient advocacy organizations, and we work to connect participants with those relevant community organizations to make sure that they have the support that they need. Family planning is also a piece of the puzzle for some families who are interested in reproductive counseling related to a genetic condition that runs in the family. And then further research. We're always advocates of further research when it's available. And we know increasingly that having a genetic diagnosis can lead to eligibility for other research opportunities like clinical trials, natural history studies, patient registries, etc. So these are all of our motivations, is helping families benefit from a diagnosis in kind of whichever way, and usually it's a combination of ways that's unique to their family and also to their specific diagnosis. So why are some patients undiagnosed? Many of you will be familiar with this, but highlighted in orange are the particular challenges that our study is designed to address. So the first one being access challenges. Not everyone is able to access genetic testing. Sometimes this is because of insurance coverage issues. Sometimes it's because of geographical barriers. They're not near an academic medical center where there is a clinical genetics team, or they can get a panel and the panel was helpful or not helpful, and they need kind of the next step in genetic testing. So access challenges is one that we aim to kind of overcome through the remote design of our study, which we'll talk a little bit more about. Other challenges that our study aims to address are analytical or technical challenges. So these are barriers to diagnosis caused by kind of the genetic testing strategy or interpretation. And what we mean by this is that perhaps just the specific test that could yield a diagnosis wasn't the one that was ordered. Or there's a genetic change that just wasn't recognized by the analysis that has already been done to date. So like I said, we have a team of software engineers and statistical geneticists who are really great at helping us overcome the technical challenges, and a whole team of analysts focused on the analytical challenges. Some patients remain undiagnosed because of what we would call a causal challenge, meaning that genetic diagnosis hasn't been identified because there isn't one there to find, because there are conditions that can be rare and can look like certain genetic conditions, but have a cause that's not tied to genetic information. So those causal challenges are not one that this study is equipped to address, because we are really so focused on the genetics in the Rare Genomes Project. Okay, so quick background. Some of you will be familiar with whole genome sequencing, but just as a quick refresher, so we're all on the same page, we'll go over what is whole genome sequencing. And to start with, what are we talking about? We're talking about your genetic information. So in all of our cells within the nucleus, which is kind of that center bit of the cell there, the genetic information is packaged into these chromosomes. And the chromosomes are composed of DNA. which is your genetic information, and then kind of the building blocks of the DNA are genes. So when we talk about the whole genome, we are talking about all of the genetic information that's contained in all of the chromosomes in all of your cells. So it's a very large amount of genetic information that is being looked at whole genome sequencing. And whole genome sequencing is currently the test that our study is doing on a research basis. So you may have heard of whole exome sequencing, and we'll talk a little bit about the difference between the two. But just to kind of set the stage, whole genome sequencing is what the Rare Genomes Project is doing. Okay, so what is it? It can be a little bit confusing when folks have had lots of types of genetic tests, and then whole genome sequencing gets brought up. It's sort of like, well, how is that different? So the difference is essentially how zoomed in or zoomed out we are on certain regions of the genome or the DNA. So there's targeted genetic testing, which you can see on the left-hand side. And targeted genetic testing is looking at specific regions of the genome, and it's usually based on the symptoms. So say a patient has seizures, we might go ahead and do a panel of seizure genes. So that genetic test is zooming in, targeting specific genes that have been related to seizure disorders in the genetic information. But it kind of puts its blinders on to other areas of the genetic information. So it's really focused or targeted there. Whole exome sequencing zooms out even further and would be looking at all of the genes in the body. So there are over 20,000 genes. It's a whole lot of genetic information. Whole exome sequencing is a wonderful and powerful genetic test. But we know that not every patient with rare disease will receive a diagnosis on whole exome sequencing. So that's where whole genome sequencing can come in. And this is what we're doing. So as you can see on the far right, we're actually not even zooming in on certain regions of the genetic code anymore. We are looking at all of it from start to finish, essentially. So this is a massive amount of genetic information. It's a bit of a double-edged sword in that we are looking at all of the genetic information. So in theory, if a condition is genetic, we should be able to identify the cause. But the flip side of that is now we are essentially searching for a needle in a haystack, and our haystack is much larger. So it can be a large amount of work on the searching side when we do whole genome sequencing. The takeaway message here really is that when you think about comparing exome and genome sequencing, exome is a very comprehensive genetic test, but even still, it is only about 1% of the whole genome. So whole genome sequencing, like I said, it gives us a much, much bigger amount of data to search through for the genetic explanation for cause of rare disease. So we're going to talk a little bit about monogenic disease. And we just wanted to kind of define this up front and make sure that folks know what we're talking about here when we say monogenic disease. So what I've shown here is kind of a little flyer that is showing causes of epilepsy as just one example. So in this example, you can see there could be brain infection or head injury, stroke, tumor, a prenatal incident, or genetic causes that could cause epilepsy. And as it says here, 50% of cases remain unknown cause in epilepsy. So when we think about epilepsy, we are assessing for just the genetic causes. So if we received an application from someone with epilepsy, we are trying to determine, is this epilepsy likely to be genetic or monogenic? Or is it possible that there's another one of these causes going on? And why do we care about this? It's because monogenic conditions, which is genes or conditions that are caused by a change in a single gene, are the ones that our study is best equipped to identify when we look at the whole genome data. So the red flags that we're looking at to help us determine whether or not something is more likely to be monogenic is if that condition has early or congenital at birth onset, If there are multiple family members affected, so if there's a brother and a sister, or multiple siblings who all have the same presentation of a rare disease. If there are multiple body systems impacted, this one isn't always true, but sometimes a rare disease that's genetic can be more likely to impact multiple body systems. We're also thinking about the severity of the symptoms. Sometimes more severe symptoms give us a stronger indication that something is monogenic. We're also looking for the absence of different exposures or environmental factors. So thinking back to the seizure example, we would be looking for the absence of head trauma, the absence of a birth injury, to rule those things out as possible contributors. This one seems a little bit obvious, but we're also looking for conditions that are known to be monogenic. Thankfully, there are a lot of really talented genetics researchers who have come before us and done a lot of research on different conditions, which has given us data about which conditions are more or less likely to be monogenic. And another example that you see here is something like obesity, where there can be monogenic causes. So you can have change in one of these specific genes that can cause a genetic form of the condition, or you can also have physical factors, chemical factors, biological factors. So those could be environmental or exposure factors. And our job really, when we see an application come into the Rare Genomes Project, is to determine whether or not a condition is likely to be monogenic, given the fact that that's what our study is geared towards identifying. I know that's a lot of information, but we just wanted to make sure that some of these terms that we use are defined before we jump into things. And I think now I'm going to turn it over to Brian, who's going to tell you a little bit more about the study itself and what participating looks like.
So thanks, Grace, for setting that foundation, providing some definitions. But yeah, all of this is the groundwork for what the Rare Genomes Project is. So we do have 4 major goals. One, increasing access to genomic sequencing. So as we mentioned, we do want to tackle folks that are not able to afford genetic testing or can't access it for geographical reasons. We are here to identify causes of undiagnosed monogenic disease for families, which Grace just went thoroughly through. We share information broadly to drive rare disease research. We know that all of this isn't in a bubble, and we are doing this and sharing our data to improve diagnoses for our patients as well as for others. And then, of course, the whole big goal of this is returning results to families in conjunction with their providers so that they may have a next step and can really take it into their clinical care. And here's just a little outline of our entire process. So it does start with recruiting patients to the project. They come in through various amount of ways, of course, through partnering with advocacy groups and foundations like NIMAC. There are clinical providers that who are familiar with us, and if they see a patient or know of one of their patients, they will send over our information. And we just actually recently launched a referral form for clinicians to directly refer patients of theirs that may need a little bit of hand-holding through the application or may not have access to internet to apply online. And then, of course, word of mouth is huge for us. We see Facebook as a huge driver of our application waves. A lot of, as I'm sure you all know, a lot of families just talk to each other and share resources. So that has always been a huge source of our applications. And then in terms of actual enrollment, so like we've been saying, the main way to get involved is to start online. We have an application on our website that allows families to talk through what's going on, the family history, previous testing that's been happening, and just give us an idea of why they're applying. We are also able to do that over the phone and go through that with families as well. Again, the whole study is remote, so we conduct a consent session for families that are eligible either through Zoom or over the phone, as can be done. And then we send them sample kits for them to get any samples drawn locally, and we work with them to get medical records. And once those samples are here, we're able to do our whole genome sequencing, and our initial analysis really is doing causal variant search through that data. As needed, we employ other analysis methods. We have access to things like RNA-seq, short tandem repeat analysis, structural variant analysis, and really do what we can being in a research setting to find things and really throw whatever we can at the data that we're getting. And again, we collaborate and share all of our data to increase the amount that we can do and help others as well. And if we do have a result, we do get back in touch with the family to work with a local provider on their end to confirm that result in a clinical lab that is part of the study as well, covered by us. And then that result is returned to the local provider, where then it kind of enters their hands to take it and go on with the next steps there. And so, of course, just to go through some eligibility criteria, we've touched upon a lot of this already, but of course, participants must be part of a family or themselves have a rare suspected monogenic condition. At the moment, we are limited to the United States. We now are able to accept families that can be consented in English or Spanish, but we do have hopes of expanding that to other languages very soon. And then having a medical provider who is involved in their care is really kind of two-faceted. One, because this is a remote study, we rely on the medical records and clinic notes for our evaluation, as well as that end result, so that we have someone to go to and make sure that the family has a provider that is willing to take this result and really include it as part of their clinical care. A couple of exclusion points. Of course, a condition that's unlikely to be monogenic, related, unsuitable for our genomic sequencing approach. And another thing that kind of comes up is for isolated cancer phenotypes. That's just recognizing that we are not the best resource for that. There are many other resources out there, and we'll generally point people to anything else that we think may be helpful for them. And so thus far, these stats are from earlier in the summer, so they're a little bit outdated, but you do see a wide range of people throughout the country. We're in all 50 states, plus DC and Puerto Rico. We're always trying to expand our outreach efforts of reaching those communities that are underserved, underrepresented, and again, maybe just far from an academic center where they have access to genetics, but we do have a wide age range. We have a pretty 49-51 even split in terms of genders. And then, as I mentioned, we're doing our best to as the whole genetics community, of improve upon this number, but we definitely lean white, non-Hispanic. And just a few months ago is when we launched and translated everything to be in Spanish. So our numbers of Spanish-speaking families is hopefully going to improve, and we have Spanish-speaking staff and all of our material translated to facilitate that. And with that, from all of those families that have made it through the sequencing, have made it through the paperwork, and have made it through analysis, here are a couple of other stats that are a little bit outdated, but just to show that for just under half of those families that have made it through analysis, we at least have a candidate gene that we are working on exploring further. A quarter total are reportable results that we work with, again, local provider and the family to get to the family. And all of this kind of ranges from being single nucleotide variants, structural variants, intronic variants. So we really see a wide range of results and hope that this number improves in the future. And then another thing that I've been mentioning kind of throughout is that data sharing and genomic matchmaking is very important for us. We recognize that data sharing improves rare disease diagnosis for our community as well as those internationally. So we do share genomic and phenotypic data de-identified through controlled access databases and various repositories where researchers can apply for access. Whenever we have a candidate gene in our analysis that maybe we want to just try and find another family to investigate that further, we just need a little bit more evidence on. We do submit those candidate genes through Matchmaker Exchange, and we are submitting variants of interest through ClinVar. And so all of this are part of the efforts that we do just to try and find a diagnosis for all of our families. And so that pretty much goes through participation and everything about our project, and are happy to open it up to questions.
And we did, I see from Karen, have a couple great questions already in the chat. Just want to make sure everyone saw those. So one of the questions was, does whole genome sequencing include mitochondrial DNA? I can't answer for all whole genome sequencing, but what we are doing does include sequencing the mitochondrial genome as well as the nuclear genetic information. And our analysis does include the mitochondrial DNA, so we are able to detect mitochondrial variants as well as variants in the nuclear genetic information. Karen also asked what the difference between our program and the Undiagnosed Disease Network is. So we get this question a whole lot, as you might imagine. I would say the main difference is our approach. Both of our teams are focused on identifying diagnoses for families with rare disease. Our approach, as you saw, is completely remote. So families will not travel to us. Everything is done, as Brian mentioned, either on the computer, the phone, email, postal mail. So it does not require really going anywhere other than to have a sample collected for the genome sequencing itself. And we are really just purely focused on the genetic analysis. So because our group does not actually do a clinical evaluation of the participant, we are not doing, X-rays, MRIs, metabolic blood work, anything other than the genetic testing. Because of this, we sometimes have families that are enrolled in both studies. This is okay with us. If we find from a family that they have previously enrolled in the UDN, we just ask for permission to contact their local site, and we'll reach out and touch base with the site there to get an update on what the analysis status is. This is just to avoid duplication of efforts. In some cases, the UDN team may be kind of hot on the tracks of a really interesting candidate that they're close to publishing. In that case, then we won't go ahead and move forward with another, whole genome sequencing and duplicate efforts there. But if that team tells us, we've done genetic testing, we haven't found anything, and things are kind of on a shelf for now, then we'll go ahead and move forward. Really good questions. Okay, let me see. Barbara, do you think that this could be viewed as a potential service model to meet the needs of those who are underserved? Yes, we hope so. I mean, this is really one of our goals and kind of our central tenets when we think about how can we reach more participants. We are thinking about who are the folks that aren't reaching us and why aren't they reaching us. So actually one of our investigators just received some NIH funding to help supplement this project, specifically looking at for families who may have a difficult time accessing this study, what are those barriers? So as Brian mentioned, that was the purpose of the referral form. So for families who can't go online and fill out the application themselves, but have a provider who can meet with them and say, I think you should do this, they can give us the contact information so that we are able to make the first contact and facilitate the enrollment with as much support as they need. So we hope that by making everything doable over the phone, through postal mail, and keeping everything remote, that we can reach more families. I'm not sure you mentioned this, but we are also hoping to expand into all languages, I would say within the next year. So we're working with our IRB right now to facilitate enrollment of folks who speak languages other than English or Spanish. And we hope that that opens up the project to a lot more families as well. Let me know if that answered your question, Barbara.
Other people have questions, either put in the chat or feel free to unmute yourself and ask a question.
Okay, another question from Karen. Do you turn down people with insurance? No, we do not. We do not ask about insurance coverage. So we are kind of agnostic to insurance and during the application process.
I will say the one kind of related nuance to that is if maybe a family mentions that they are pursuing clinical testing at the same time as applying to our project, we will probably put them on pause, just of course recognizing that clinical testing will be much faster than doing research, whole genome sequencing, that if they do end up getting it covered, we'll either wait for them to see the result, or if they're just satisfied with that, we'll let them do what they would like to do. But then, of course, if insurance comes back and it's not covered, or they do end up having the test and it's negative, we will be happy, I think, to accept them into the study.
Yeah. So as long as no one is pending, genetic testing that is in the lab and coming back soon, or they've submitted a request to have genetic testing that they need approved. If there's something really actively in the works on the clinical side of things, we'll just wait until that's moved forward and then go from there to determine eligibility. But yeah, that was a great explanation. Karen, the $1 million question. What is your turnaround time? So in the research space, this is almost impossible to define. We estimate that from the time that we get blood in the lab from enrolled families to the time that our team has the whole genome sequencing data and is able to begin their analysis can be about 4 to 6 months. From there, the analysis duration is kind of open-ended. So for families who have actually a pretty clear diagnosis, where it's a pathogenic or likely pathogenic variant in a known disease gene, and their trouble getting a diagnosis was more on the access side than the analytical or technical challenges side, that result can be back to the family within the year. But for families, especially those who have had clinical exome before, because it's such a powerful genetic test, and tends to find those really clear-cut, low-hanging fruit diagnoses, at that point, really the turnaround time can be indefinite. We have families who have been participants for multiple years who are receiving results, this month. I am currently returning a result to family number 78, and we're up in the 2000s now. So we don't necessarily have an end date, but I think the nice side about that is it's because we continue to reanalyze families when we have not found a diagnosis after the initial analysis. Great questions coming in. Okay, in addition to the link on the website, does RGP have a one-page flyer or something similar which describes the project and could be shared? You want to take that one?
Yeah, we do. Maybe we can definitely share it with Alyssa and the NIMAC team, and maybe they're able to distribute it there. Sometime soon, we are hoping to do a website revamp where we'll have all of this material up there as well. I know we have just our information there, but having actual flyers on there is something we plan on doing. And then, of course, feel free to reach out personally, and we're happy to share any of our I guess, recruitment materials with you as well. So just to mention, for anyone that, if that's easier for patients, we also have a YouTube video that outlines participation that is available on the website if you're on the, I believe, the How It Works page, or if you just look up on YouTube, Rare Genomes Project, it'll be what comes up. So definitely can share things that can be shared with others.
Yeah, and we do have those flyers are available in English and Spanish as well. So we can make sure we share both of those. Another question here, do you look for repeat expansion disorders? Yes, we do. So our analysis is looking at single nucleotide variants, structural variants, like deletions, duplications, insertions. We are looking at the mitochondrial genome. We are looking at repeat expansions. Am I missing anything that you can think of? That's the beauty of genome, is that we're really able to detect a whole lot. Epigenetics, ooh, good question. I want to say no, in that we are not doing a separate analysis, but of course we are looking in kind of the regulatory regions, given that we are looking at genome, but we are not doing kind of like separate methylation analysis at this time. Okay, a question about reanalysis. Yes, we do reanalyze. So for families who do not have a diagnosis identified at our first analysis, they do get reanalyzed approximately annually. Of note also, some of the, sometimes we have like a new method of analysis that gets added. So for instance, when I started with the project a couple of years ago, we weren't able to look at the mitochondrial genetic information. That was a strategy that was added. So when that was a new analysis strategy added to our project, we did it retrospectively. So anytime there's a new analysis strategy added, every family that is unsolved gets that retrospective analysis method as well. I'll let you take this next one, Brian.
So yeah, good question, Alyssa. Do you typically get samples from just the patient or also from biological parents? The answer is anything that we can get, honestly. Death trios are definitely ideal, but we definitely have a lot of older patients where we just have themselves. We are also able to enroll affected other family members, so siblings, if there's an aunt or uncle, or occasionally if there's a child that's affected, we'll enroll them as well. But we typically get samples from trios when we can, or just the patient or just one of the parents.
Yeah, and when we can is kind of the key there. So trios make our analysis easier. And for those who aren't familiar with the term, trio is a child and then both biological parents. So it's best for our analysis team when we have DNA from all three, but it's not required. So we do have participants who are adopted or who do not have contact with one of their biological parents, and that's okay. We don't determine eligibility based on which family members are available. Do we accept saliva samples or other alternatives to blood? So we really strongly prefer blood. It gives us the highest quality genome sequencing data for our analysis. We have made a couple of exceptions over time in permitting saliva with a little bit of a caveat that the data quality may not be quite as good as it is with blood. Anything else to add there?
I just know that that's something that I think might be improving soon,
That's
but
fair.
yeah, I think pretty much said it, that the strong preference is for blood. And then typically if we do have to move to saliva for another reason, we'll kind of work on that family by family.
Of note, we are able to enroll individuals who are deceased. So if, for example, a patient passed away and the family is still kind of searching for a genetic diagnosis, and that individual had clinical genetic testing, and that clinical laboratory has a stored CLIA DNA sample, we are able to request stored DNA. If it's someone who is living and able to provide a new sample, we would like a new sample, but in cases where there is genetic information available, stored, or DNA specifically that's stored clinically, in certain cases where that's necessary, we can obtain a portion of that sample. Okay, a question from Alyssa. Is there any minimum amount of evaluation that you'd expect a patient to have before enrolling? Do they have to see a genetics professional, have a karyotype? Really good question. So the short answer is no. There's no specific evaluation that we require. This goes along with our interest in making this project available to folks who have trouble accessing clinical genetics care. The kind of caveat there is that if someone, for example, had a clinical diagnosis of a genetic condition that has targeted testing available, for example, neurofibromatosis, we would strongly recommend that they try to pursue clinical testing first if they have not tried, just because it is so much faster. And because genome can be a little bit of overkill in those cases where targeted testing has a relatively high diagnostic rate. So in those cases, we can actually generally kind of work with the family to connect them with a genetics professional locally or telehealth genetics to see if they can go about getting the targeted testing. But if that's not the case, then yeah, we don't require a specific evaluation to be done in advance or any particular genetic testing to be done. But similarly, if someone is, just started their clinical genetics journey and they saw a geneticist and they ordered a microarray and karyotype, but they also talked about genome, we would recommend that they follow through with whatever clinical genetics eval is ongoing, and circle back to us once that testing has completed. Good questions. Karen, I see your question. How about newborn blood spots? Good question. At this time, we are not able to do whole genome sequencing from newborn blood spots.
I'm curious if you're getting families from the NICU. Are you getting families that young, patients that young, or are they usually kind of a little deeper into their odyssey before they get to you?
Yeah, we certainly have very young neonate participants. In some cases, it's families where they enrolled with one child with rare disease and then had a additional child who presented in the neonatal period or even prenatally. So in some cases, that's kind of how a very young participant will come to us. We do also, one of our team members is a neonatologist geneticist who is seeing her patients, and in some cases, they'll do the clinical diagnostic workup that's available to them in the NICU. And then, she sometimes refers cases to us. So yes, I would say we're not really frontline genetic testing for
Yeah.
NICUs. And research testing isn't designed to be frontline anyway. But yes, we do have some, what's our participant age range is like days to 90 something years. So we have a really tremendous age range.
Yeah.
Okay. Are you able to share the clinical lab that you use for WGS? Good question. So we are actually we're doing research whole genome sequencing here at the Broad Institute. So the whole genome sequencing is happening here at Broad using, we have all Illumina next-gen sequencers here at Broad. And the clinical confirmation of the result happens at a couple of different labs depending on which methodology is needed. So we work with the Laboratory for Molecular Medicine, which is the Harvard Partners Healthcare Lab here in the Cambridge, Mass area for some clinical confirmations. We worked with GeneDx for a couple of confirmations. The Mass General Hospital also has a clinical lab that we've used for a couple confirmations. So just depending on which technology is needed to confirm the research result, we'll work with different CLIA labs as needed. Good question. Great.
You gave us the example kind of early on today of epilepsy being one kind of symptom that brings people to you. And I also saw in your notes that there are certain hereditary cancer conditions that aren't a good match. I was just wondering if you could give us some other general categories of common things that bring families to you, just so they can have a sense of what is kind of, I don't want to use the word appropriate, but what do other families kind of end up coming to you? What kind of symptoms?
Another big, I guess, cohort of ours are folks with like neuromuscular conditions. So they've been diagnosed with like general muscular dystrophy with kind of an earlier onset than usual, or they've had the genetic workup already and whole genome sequencing is the next step for them. So that's definitely another major group that we see.
Yeah, I would say kind of the syndromic neurodevelopmental disorders is a very significant category. Let's see, other neurological presentations like ataxias, epilepsies. We have some kind of hearing loss, vision loss families, craniofacial conditions,
few kidney.
Yes, some nephrology patients, some rare kidney diseases. But we really are open-minded in our review of applications in terms of the phenotype or the symptoms, as long as it really kind of checks the box on having a reasonable likelihood of having a monogenic cause. I see a question here from Lynn, who mentions that her family has been through quite the diagnostic odyssey and has had prior genome studies through other research groups with no diagnosis. And the answer is yes, we do still consider families who have had similar testing, but no answers. So it's all case by case. But yes, we would encourage you to submit an application if you'd like to be considered for the project, and just give us as much detail as possible, and we'll go from there in terms of determining eligibility. And sometimes we may reach out with some follow-up questions if we need a little bit more information about things like that. But that is not in and of itself a reason that we would not accept a family. We do have a number of families who have been on, many year-long, decade-long diagnostic odysseys, who have been involved in other research projects as well. Hope that helps. I'm an open mic, great. Well, I do review all applications that come in, so I'll keep an eye out for it. These are awesome questions.
Yeah. And I know we're running low, starting to run on low on time. So of course, feel free to reach out to us if a question comes up later. If you didn't want to ask a question in front of the group, we are open to answering all questions as well on our end.
Anyone else have other questions today?
If there's any providers out there who have families that come to mind, but you would kind of like some feedback on possible eligibility before referring them, you can shoot us an email. We have a number of providers who, I think sometimes like to get a little bit familiar with the project by asking about potential eligibility of a couple families, and then can kind of get a sense of the families that are best for referral. And so we're happy to give some feedback there about families. You also don't have to do that. You can go ahead and recommend families apply without contacting us. That's most folks get to that point, even if they do start with kind of the email consultation first. Emma asked how many genetic counselors are involved in RGP. That's a good question. So myself and Melanie are both genetic counselors who are full-time with the Rare Genomes Project, but our group does involve other genetic counselors. So we have one of our analysts is a genetic counselor. We have a team member who's in charge of data sharing for the group who's a genetic counselor. And we also work with the matchmaker team and the ClinGen team, and a lot of that work, that group's projects are managed by a genetic counselor. I'm forgetting. We're growing, it's exciting. Oh, and then we just actually recently have 3 genetic counselors hired on the laboratory side as well. And we often have students rotating with us, which we love. So we're small but growing in genetic counselor representation over here.
Great. Can you tell us a little bit more about what happens when you do feel like you have something to share back with the families? Is it the counselors that reach out to the families, or what does that look like?
Yes, it is. So either Melanie or myself contacts the family, sometimes reminds them that they're participating if it's been a little while.
Yeah, sure.
And we will tell the family, we found a genetic result that we think explains all of the symptoms that we're aware of, or explains at least a portion of the symptoms we're aware of based on what the fit looks like to us. And we do give them the option of moving forward with clinical confirmation. No one has said no so far, but it is an option should a family have changed their mind about learning about the genetic diagnosis in the interim. And then we ask them which provider they would like us to work with to return the result through. We do generally strongly recommend that if they have a genetics team that they've seen in the last handful of years, that it comes through a genetics clinic, just because we know that these are going to be the professionals who are best equipped to support the family through the diagnosis. But we've also returned results through plenty of neurology teams, nephrologists, primary care physicians in some cases. And then for families who end up, sometimes kind of in a lurch where they don't have a provider on their team who is comfortable returning a genetic result, we have partnered with telehealth genetics providers to make sure that they get the support that they need through that return of results process. So I think about it as kind of like our job as genetic counselors in this role is actually a little bit more to kind of counsel the team up front, especially if we're working with non-genetics providers to return the results. But we are here to support the family and the clinical team in any way that we can throughout the process. And we do follow up with families after the result has been returned to assess need for other resources or connections. But we do, we really want to make sure that the transition from the research space to the clinical space is done as smoothly as possible so that that genetic result can be integrated into their clinical care.
Great. And have you had families come to you that already have an existing genetic diagnosis, but there is suspicion that there might be a second one? Some of us have heard about those families that, come up with two things. I'm wondering if you're seeing that come to you.
Yeah, we have. I would say it's not super common,
Sure.
but we have had a couple. In some cases, what we're actually able to determine is happening is some type of a communication gap where there actually is a diagnosis that really explains everything. I'm thinking of one particular family that had a rare genetic condition, and the family applied, and it took us a little while actually to sort out what wasn't explained. And in talking with the mother, it turned out she was really concerned that the child was having recurrent ear infections that weren't explained by the diagnosis. And so we were able to kind of discuss with her and the clinician that, not everything, may be encapsulated by a single genetic diagnosis. And sometimes kids have run-of-the-mill, kid conditions as well that aren't connected to the rare condition. But sometimes we have a couple of families who do have a genetic result that, for instance, I'm thinking of one where it's a child with a complex presentation who has a pathogenic variant in a hearing loss gene and does have hearing loss, but definitely has more going on than non-syndromic hearing loss. So that family was enrolled to see if we could identify the cause of other symptoms there. So yes, that does happen.
Great, thank you. All right, so we see we're about at time. If anyone has a last minute question, go ahead and put it in, but I'm sure a lot of people want to follow up with on your website and the email that you put there with other questions. And, we know that we come across a lot of families and communities that are really struggling to figure out how to continue on their diagnostic journey that they've already been on. So we really appreciate you sharing these resources. And thank you to all the families and advocates and professionals who joined us today also.
Thank you for having us, and thank you for all that you do for families. It's great to connect with y'all.
Thank you all.
Thanks, everybody.